at how microscope technology has improved and how with these improvements we can see with a lot more clarity intracellular structures, which are the subcellular structures in cells. We're going to look at the numbers involved with size, scale, things like resolution, magnification, and the prefixes milli, micro, nano, and pico, which are really important and often students get wrong. The first microscopes came about in the 1600s, and they have a really simple design. An eyepiece lens, which is the one that's looked through, an objective lens at the bottom, a light source, and a sample holder where you put a specimen over and shine up a light. The light travels through the objective lens into the eyepiece lens and into the eye. And that hasn't changed. So on our modern microscope or a modern light microscope, we've got a mirror here to reflect the light, or we can use a light source, reflects it up through the stage. So this part here is the stage, through the specimen into the objective lens and up into the eyepiece lens and into the eye. To calculate the magnification of any light microscope, you need to know two things. Firstly, you need to know the magnification of the objective lens, so that's this one here, and the magnification of the eyepiece lens. And really simply, you multiply them together and you get the overall magnification. So for example, if we have an objective lens, so this lens down here, of 40 times, and an eyepiece lens of 10 times, our overall magnification will be 40. So here on our objective lens 40 times by 10, and that will give us a magnification of 400. Two key words you must be really conscious of when talking about magnification, especially when it comes to comparing light microscopes and electron microscopes. The first one is resolution. Now, resolution is the smallest distance between 2 points that you can see as a clear point. So when we talk about a high quality image, what we're actually saying is that it's a high resolution image. And you can see here on this diagram below, we've got clear individual beads and this blurred low resolution image. Now, the other word you need to be familiar with is that of magnification. Magnification simply means how much bigger something looks compared to what it actually is. So this picture of a bird of prey here, looking at a long distance, it's under a low magnification, it looks quite small. If we increase the magnification, it looks a lot bigger. So some specifics of the light microscope. Firstly, what it does is in its title, it uses light. So a key part of it uses light to illuminate the specimen. It can magnify quite a lot, up to about 1500 times, so that can see the nucleus of a cell. Can't really see anything more than that. So the smaller organelles, such as the ribosomes and the mitochondria, you won't be able to see with a light microscope. And as we looked at already, it takes two lenses to magnify the sample, and you have to stain the specimens. And if they're plants, you tend to stain them with something called iodine. And if they're an animal cell, you'll tend to stain with something called methyl orange or methyl blue, which just make it easier to see under the light. Electron microscopes are a much more recent invention. They use electrons to make an image. So in the same way that with a light microscope it passes light through a sample, electron microscopes fire electrons through a sample. Because electrons are so small, it gives a very, very high resolution image, much higher than that of a light microscope. The problem is with it, it's hugely expensive, takes a lot of skill to use, the specimens have to be dead, and you can only get black and white images. You do see coloured ones, but they're coloured artificially after the image has been taken. When working with microscopes, we're talking about numbers that are really, really small. So rather than talking about things that are 0.003 of a millimetre long, we use other units, and the units we use tend to be micrometers and nanometers. So when we're talking about the organelles in a cell, or cells themselves, we're usually measuring them in micrometers. Now, a millimeter is 1,000 times smaller than a meter. So 1,000 millimeters will fit into a meter. A micrometer is 1,000 times smaller still, so that means 1,000 micrometers in a row would fit into 1 millimeter. A nanometer is 1,000 times smaller again, so that means 1,000 nanometers in a row will fit into 1 micrometer, or 1,000,000 nanometers in a row will fit into 1 millimeter. And so you can see now we're starting to get to tiny, tiny units, which is why we have to use these prefixes. For example, a cell is 30 micrometers long. How big is it in millimeters? Now we know that 1 micrometer is 1,000th of a millimeter. So all we have to do is take 30 and divide it by 1,000, and that will give us our answer in millimeters. So the answer will be 0.03 millimeters. If we look at the same distance or the same size and work it out in nanometers, we go the other way. So we know that there are 1,000 nanometers in a micrometer, therefore we have to times that 30 by 1,000, and we'll have 30,000 nanometers is the equivalent to 30 micrometers or 0.03 millimeters. You may get an exam question that looks something like this. State 2 advantages of using an electron microscope to view cells rather than a light microscope. This is a really nice question, and all it wants you to do is give a couple of the reasons discussed in this video about the benefits of electron microscopes over light microscopes. So you'd start by saying an advantage of an electron microscope is a much higher resolution image. That will pick up 1 mark. Because it's requesting 2 marks, it's a 2-mark question, you need 2 good points. The second mark could come from either there could be a much higher magnification, or you could say you could get a 3D image. Any of those points will be enough for the 2 marks. Try testing yourself on what's been learned. Pause this video and have a go at these questions. I'll put the answers in the comment box on YouTube.